So our next speaker, it's a pleasure to introduce Dr. Juergen Liebermann. Juergen has been involved in IVF since the late 90s, or mid-1990s, sorry, not the 90s. He did both his initial degree and then his PhD in Bavaria, Germany. So you'll hear a bit of a German accent, I think, even though he's been in Illinois, in Chicago for a long time now. I think he's been director of the fertility clinics in Illinois for a long time, and in the mid-2010s, 2017, he became scientific director of COPS, which is the US fertility group now, which is the largest IVF conglomerate, I think, in the US at the moment. Juergen's incredibly well known for the adoption of vitrification, modification of the vitrification system. He's published extensively in the area, has some reference books that nearly all embryologists look at in terms of vitrification. And he's still leading the way in doing modifications to vitrification and freezing technologies. And I think he's been doing the lecture circuit for a while now, because he's developed a technique, and it's a brilliant introduction to fast and furious, rewriting vitrification warming protocols for human oocytes. And I can tell you, coming from a very busy lab, this has sort of revolutionized the way we do warming after vitrification. I think vitrification really changed the picture, I think, in IVF for a lot of us. But now the new warming protocols that Juergen's been very involved in redeveloping, I think have changed a lot of things. So it's a great pleasure to introduce Juergen. Thank you.
Thank you very much for this very kind introduction, Danny. And thanks, Nabil, for inviting me back. Last year, I talked about stuff what I just started to look into this year. I can give you data based on peer review papers, and I will talk about two papers I recently was able to publish in RBM Online. One is about blastocysts, and one is about oocytes. So you see, I have only one thing to disclosure. And you see here my talk outline briefly on blastocysts on the left, and then I will move on to oocytes. And when I started this research, just to get a little bit of time frame, I started in May of 2022, and then my first breakthrough with oocytes was in November of 22, and then publications happens actually much later. So it takes a while, over 2 years, to get it finally published, with also the time you have to consider for the reviewers. And if I disappoint you, I feel sorry, but we will keep freezing until the end of our days, because when you look at these numbers, there is no way that we will have less cycles or less patients dealing with infertility. It's actually the opposite. The numbers going up based on the SAR data I rely here. So whatever you look, is it fresh number of cycles, is it cycles with own eggs, donor eggs, or frozen embryos, everything just went up every year. So we will keep freezing. And this is also true just recently, new data comparing 21 with 22, again from SAR, preliminary released. And you see also here a number of cycles, in fresh cycles about 6%, and for freezing 21%. And when we look to our colleagues in Europe, last year, Copenhagen in Escher, so they estimated about 30% of all embryos from IVF are frozen embryos, from frozen eggs or frozen embryos. And this is a number because, as Danny pointed out, I worked with freezing. I have only 56 seconds left. Because I freeze really long enough, and we always had like 20%. And now in a sudden, we see that jump up to 30%. There's a couple of reasons, right? It's also involving PhD that many of us do to maybe sometimes 100%. So we definitely have an increase of freezing cycles. So now is also time, that we have to think about, do we still do that after so many years of vitrifying? Do we still do the right thing? I started looking into vitrification in 2000, and since then, barely things have changed. Same solutions, same time frames. We changed a little bit the way how we do embryos equilibrate by not combining drops. We make single drops, and then we put a time on and let them sit in the solutions. And so there was time for, there's always people, they already do some good homework. And there is no point here not to mention Thiel, because he had some prize papers on this at PCRS and at ASRM. And so he, by accident, dropped the embryos in 1 molar sucrose for 1 minute and then published that. So, and then, when I read this, I take it home and do my homework, right? And so we have plenty of embryos. They are designed for this position. We are a big clinic. We do a lot of cycles every year. Now for 20 years, I'm with FCI since 2004. There's a lot of embryos we are able to use for donation. And we can use them for training in-house embryologists, or even for doing something like this, and also for stem cells. So it's a patient decision what they like to use their embryos. So, and these embryos were for this position. And as you see, when I started in May, I fought with help of one of my supervisors, 564 blastocysts. First, when I started, I want to have 50. And then I said, and now I need 50 more. I was at 100. Then we went to 200, and there was a question, Juergen, do you think we have enough? I said, no, we need more. And because N numbers are strong, right? And when I had the 564, I said, that's enough. And that convinced us to present that to the physicians. And so in September of 22, we made the conversion to this protocol, right? And what I also found when I fought with embryos, I separated them between BGT embryos and non-biopsied embryos. And what we found is very interesting that, overall, when I mix them, about 76% were able to hatch out after 24 hours. But for the biopsied embryos, it's more than 87%, whereas the non-biopsied embryos were only able to hatch about 60%. So we would lose about 25% if we would do not assisted hatching. So for me, as a learning curve here, we still would apply at least for the non-biopsied embryos assisting hatching, just as a side effect. So this is our freezing protocol. We freeze blastocysts in 5 minutes. Everything is at room temperature. No, I don't do 37. We have one drop we rinse. When we put them on top of this drop, it's ES. For some of you who don't know, it's called equilibration solution. It contains 2 cryoprotectant at 7.5%, so a total of 15. And after 4 minutes, we move the embryos in these 2 drops, keep them here for a remaining of 60 seconds. This is now a double concentration. It's now 15% of both cryoprotectants, so it brings it to 30%. And after 1 minute, we put them on a carrier, plunge them, and store them. So freezing here, a total of 5 minutes. And when we come to the warming, it takes us now 1 minute, right? Before it was 11 minutes. What we do now, we have one single drop, 400 microliter of TS. This is 1 molar sucrose. And now at 37 degrees, everything I do now, warming, is at 37. And we keep them for 1 minute, and then they go into transfer dish, and we are ready to transfer them. So overall, I estimated a couple of embryos here when we transferred them. The maturity of embryos, about 80% are healthy looking, full expanded at the time of transfer. It's about 2 or 3 hours after we warm them. And there's also some, they don't make a good impression, but still good enough that we have a good feeling to transfer them, because on the end they still make a good outcome, but this is only 20%. This is our up-to-date numbers. Just a couple of days I updated them. The last transfer day was May 18, so 2,700 transfers. And you see what you can expect, or positive beta per age group. It's consistently over 70%, and there is a drop for patients older than 40 to 65%, so in average 72% positive beta. And I want to quickly go through these numbers because I have a time constraint here. You see here what you can expect on ongoing pregnancy and implantation per age. You see here per day of development. We talked about this, right? So you definitely get a significant better outcome with day 5 than day 6. Implantation also better. And when you go to the untested and tested, when you look at these numbers, you may be a little bit disappointed, don't see the benefit of PGD. But if you look at the age group doing PGD, for example, here, 38 to 40, you see non-biopsied embryos versus biopsied embryos. And here, non-biopsied versus non-biopsied embryos, you see the benefit in older patients for doing genetic testing. And I want to move on to the next level, that would be live births. And so far, we have 800 and close to 900 transfers, and we have 460 babies born with a live birth of close to 52%, normal birth weight, normal duration of pregnancy, and also a high number of singleton and only 2% twins. You see a little bit a difference here. That's older numbers from 2007 to 14. You see 50-50, but here you see a difference in gender, 43 versus 48, 47, and you might know where it comes from. This is all based on sex selection. So to summarize the blastocysts until then, and I have 16 minutes, I can make it. It makes laboratory technique for warming blastocysts simpler. It reduces the unnecessary time blastocysts are exposed to room temperature, and in turn reduces the stress to the blastocyst. There is less time for radical oxygen species creation and their negative impact, and you have them, on the embryo development. I also tested out, because of time, I didn't show, lower sucrose concentration, they work as well. And we usually, when we have a transfer medium for embryos or oocytes, we culture them for oocytes until ICSI in 20%, 10% works as well. And you wonder why I did this. I did that for European colleagues because they cannot add protein to the media. They cannot alter. They don't even have 20% protein media. So to give them also that protocol, I tested the 10% and it works with 10% as well. Short dehydration, short rehydration is easy to implement in your daily routine and standardize your blastocyst cryopreservation procedure and its outcome even more successful. The one-step warming protocol is superior to the multi-step protocol in terms of pregnancy outcome, efficacy for supporting an optimized workflow, and is reducing labor as well as cost-effective. And that's my personal calculation. When you come down to the time we would save here to the old protocol and also on in terms of consumables and media, that's my calculation what we would save. Now to the oocytes, and oocytes here, it's very different to freeze when blastocysts, and I'm sure most of you know when you can freeze blastocysts, don't expect you can also be so successful with oocytes. But oocytes also is something we will deal with more than ever. This is just a number from us, and you see the trend in terms of our numbers of egg freezing every year. We now add more close to 500. You see here what happens when ASRM removed the experimental label in 2013. There was an even more increase, 5-fold increase in freezing for us, and I'm sure for many of you. And you see also when I post those on LinkedIn, when we talk about eggs, people are hungry for. You see how many impressions and how many people give their hands up, usually not hands down, and also a lot of comments. So there is still a demand. Even we freeze now for 20 years, we vitrify for 20 years, we freeze eggs much longer. First egg freezing was reported in 1986 by Dr. Chen from Australia, first live birth. It was never repeated by anyone else. But the first publication on oocytes vitrified with live birth was in 1999 from Dr. Kulusheva. She reported that. So when we talk about eggs, there is something missing, right? And when we look at the puzzle, I would say there was a puzzle we were missing. And again, I was falling over a publication that is, and I have a very good relationship to Miguel Gallardo, who lives in Spain. They published this in 2019. And what they found is that the oocytic osmotic behavior indicates that the dehydration upon exposure to the standard cryoprotectant happens very fast. The point of minimum volume of the shrink bell curve is reached in 60 seconds. At this point, intracellular water ejection is completed, which coupled with the permeation of the low molecular cryoprotectant results in similar intra and extracellular solute concentration. This is what you want to achieve. When you dehydrate cells, you want to reach an equilibrium, you want to have a state of isotonic conditions where osmolarity is inside and outside. So according to them, it happens really fast. That means that prolonging exposure to the cryoprotectant solution does not improve the cytosolic class forming tendency we need to vitrify successfully. After 2 minutes of exposure to standard cryoprotectant solution, the critical intracellular solute concentration necessary for successful vitrification was attained. So, I started in May, and actually in November of 22, I had that protocol, what I now presented, what I published. But it took me 5 months because I was so focusing on using the solutions we use all for so many years until I tried 0.5 molar. And most of you know when we 4x, we go in 1 molar, and when we dilute the sucrose, that's the standard way. It's a dogma in cryobiology that we have to reduce the sucrose concentration to gently allow water to re-enter the cell. And so I started with 0.5, and that helped me, to get my breakthrough. And so I have 1 drop. First of all, that's a vitrification. I have 1 drop of VS for rinsing, 1 drop of ES. So I put the X on top, let them sink. After 1 minute, I move them in 2 drops of VS, overall 1 minute. So in 2 minutes, I vitrify the oocytes. 1 minute here and 1 minute here. And again, this is ES and this is VS, 7.5 of 2 cryoprotectant, and again here 7.5. And when I'm done, usually our old protocol was like 16 minutes, right? Now I do it in 4, and this is my warming. And I start at 0.5 molar, and then I go, and I have a bigger volume. I make like 500 microliter. When I go in 2 drops of WS, I always carry sucrose over in here, right? And then 30 seconds later, I go in the final drop to release every sucrose I might carry it up in this drop, and then they go in the culture dish until ICSI. So thawing 2 minutes, 1 minute here, 1 minute here. Before, it took us about 10 to 11 minutes. And so when I started material I can easily use because our patients signing, when they have my treatment plan, they're signing for the eggs or embryos. We don't cryopreserve, we don't use for them immature oocytes, and so we can use for research. And so I started with German vesicle and metaphase 1. They didn't convert until time of ICSI. And you see here, I tested 2 different media. I tested more, again, because of time, I can only show these 2. And I had here 311 for Irvine and 348 with Vitrolife. You see their survival, and you see here's a metaphase 1 and in both media and their survival. So consistently over 90%, 2 minute vit, 2 minute warm. And of course, I don't stay there, I keep them overnight. And what I do, I run also a control. I did also GVs, not vitrifying in overnight in culture to see how they convert to metaphase 1 or metaphase 2. So you see here my GV vitrified, you see here's a fresh one, you see their conversion to metaphase 1 close to 97.2%, here about 89, close to 90, there is no significance, that was so non-vitrified. And this one here is a conversion of this vitrified here to metaphase 2, about 56%, and you see here's a metaphase 2 from fresh CVs converting to a metaphase 2. And when you look at the metaphase 1, the vitrified converting to 85% to metaphase 2 24 hours later. And when I use fresh ones, they didn't use for ICSI because we were not ready for ICSI. They converted pretty much to the same percentage 24 hours later when the vitrified metaphase 1. And then I want to show you a nice study just recently published, and I just made these slides 2 days ago. And they compared here 2 protocols. They had oocytes that were fresh, not vitrified. Then they looked in oocytes were vitrified, conventional protocol that was 10 minutes in ES, 1 minute in VS. And then they compared here ultra-fast vitrification 1 minute, as I showed you before, in ES and 1 minute in VS. And you see the survival, they considered the non-vitrified 100%, obviously. And when they related this to the 100%, you see there is equal survival. There's even a higher survival when we do ultra vitrification. When they looked at different intracellular patterns, for example, they looked at the endoplasmatic reticulum, and they graded them as normal or as abnormal. And when the cells are here distributed in the equatorial part, they call that normal. If it's abnormal, we have small areas of endoplasmatic reticulum. And when they looked at the presence of clusters containing inositol, inositol is a structure very important for cell membranes. When they were related outside, they call that normal. If they were not existing or sporadic clustered, they call that abnormal. And when you see here the analysis of the data from the fluorescence, you see the fresh oocytes, you see conventional VIT, and you see ultra VIT. And you see the ultra VIT is very equivalent, comparable to the non-vitrified oocytes in regards of the endoplasmatic reticulum. If you look at the presence of clusters, there is much less here in the ultravit. You have more when you conventional vitrify. Again, ultravit very comparable to the fresh. And when you look here at the intensity, there was no difference in all three groups. When they looked at the mitochondria and the membrane potential as a key indicator for mitochondrial activity, and they considered here again normal versus abnormal. If they even distribute, they call it normal. If they have aggregations like here, then they call it abnormal. When they did a fluorescence on the mitochondrial membrane, and they call it normal if it's green or red, but if you get both color mixed, they call it abnormal. And you see here the mitochondrial membrane potential results from redox transformation associated with the activity of the Krebs cycle and serves as an intermediate form of energy storage, which is used by ATP to make ADP. And again, here's the data analysis. You see here, even distributed in a lot here in the non-vitrified, much better here in the ultravit compared to the conventional vit. If you look at the intensity here, green, red, you see much less here comparable, actually not very different between both. And when we look here at the membrane intensity, there is also here this one, the ultravit, very comparable to the fresh, whereas we see here less intensity, more mixed up in the conventional vit. And when we look at microscopy, scanning, und zwar spindle of the mitotic spindle and the chromosomes, you see here nice pictures between spindle and the chromosomes, and you see here the comparison. There is also no difference between these two different ways how to vitrify. There is a little bit a slight abnormality, but it is really not significant between conventional and ultravit. And that's fun. It's good to know that we didn't compromise X in that regard. And I have 2 minutes left here. And then we show here the comparison of oocytes volumes excursion based on the cryoprotectant. You see this is the ultravit, right? So you go here 1 minute in ES when you dip them in VS. So you stay pretty much at the same volume. What happens here in the long protocol, you go to about 10 minutes or 15, because after volume loss, the egg will recover, it will re-expand, and then you shrink it again, right? And this is definitely a problem later on for the oocyte development. So you don't have these extreme changes in volume here compared to here. And to come to my summarized, they also have fertilization. You see here, fertilized between conventional and ultravit. There is also a higher percentage. You see also cleavage is also higher than the conventional. And when you go to the blastocyst formation, they are equal to the fresh one, whereas the conventional vit, the longer protocol, drops in the number of blastocyst formation. And to summarize their paper and overall, the ultravit protocol with minimal volume changes exhibits a similar reticulum distribution pattern to the control group and did not experience negative effect on embryonic development, unlike the conventional VIT did. During reduced volume changes, the concentration process, which is necessary to eliminate intercellular moisture, led to microtubule damage. However, ultravit results in less damage to mitochondrial distribution owing to smaller volume excursion changes. Conventional VIT method may cause potential damage to crucial cell organelles in mouse oocytes due to cryoprotectant toxicity and osmotic stress. And finally, a 3D image showed that conventional VIT and ultravit retain over 98% of the normal morphology of the meiotic spindle and chromosome. So no difference here. And I also want to show you clinical data from an egg bank on using the same protocol I showed you in terms of fertilization, in terms of cleavage, blastocyst formation, and then the rate of normal embryos. This data will be presented at Escher in Amsterdam. He did also metabolic studies, and where clinical pregnancy, freezing eggs in 2 minutes and warming in 2 minutes is 65%. And finally, what leads me to do all this, and I think this is also a theme for you and your meeting. Now, Bill, it's a saying from John F. Kennedy: change is a law of life, and those who look only to the past or present are certain to miss the future. We definitely here, all in that room, don't miss the future. So thank you very much for your attendance and listening to me.